So with the evolution of PCR, obviously we've been working very closely with our research and development team to create new enzymes for being able to address challenges that customers may face when doing these types of reactions. So the course of this discussion will first start talking about the work that we've done in the area of polymerases. And then in the second part, we'll actually take the information that we've gotten from this research and apply it towards some specific experiments to show how we've been able to address some of these challenges. So first, we'll start with precision. So when working with PCR, in some cases, the fidelity or accuracy of the polymerase is very important, but not in all cases. So when you're working in an area where you're just doing genotyping, or you are looking for the presence or absence of a sequence, or you're doing colony screening or something like that, then you may not actually need a high fidelity polymerase. But then when you're working with experiments where you might be cloning, you're doing some sort of pre-amplification step for next-gen sequencing, or you're doing site-directed mutagenesis, or any kind of SNP analysis looking at variability, then obviously the accuracy of the polymerase is very, very important in those experiments. There are three specific characteristics of DNA polymerases. The first is the 5' to 3' polymerase activity, which is actually what generates and synthesizes the strand of DNA. So the DNA binding domain binds to the template strand and actually progresses synthesis by addition of bases in the 5' to 3' direction. All DNA polymerases exhibit this property. The second characteristic is a 5' to 3' exonuclease activity. What this does is, as the DNA polymerase is moving along the template, if it encounters any thing that may also be bound to the template, it will actually hydrolyze that and release it so that it can continue its synthesis strand. Typically, the way TaqMan chemistry works is that a probe is bound. As the polymerase moves along, it will actually encounter the probe, hydrolyze it to release it, and you get fluorescent detection, which is the common way that these probes work in qPCR. The third activity that can exist is a 3' to 5' exonuclease activity. What this does is it's actually going in the opposite direction of strand synthesis, and it's looking for any potential errors or mistakes that the polymerase may have introduced, and it will actually cleave any mismatches and make that correction. It's commonly also called proofreading activity. So some proofreaders that are in the industry will only have this activity, and some it may be reduced in some capacity. Other attributes that are obviously important in PCR are the thermostability of the polymerase, the accuracy, speed of the enzyme, as far as especially when you're creating longer DNA templates, specificity, processivity, and inhibitor tolerance. And we'll talk a little bit more about these in the next slides. When you're talking about fidelity, you're actually talking, as I said, about accuracy. And in the industry, there are some common ways that you can measure for the error rate of a polymerase. The first and probably most commonly used method is to do a colony-based screen. where you do a PCR reaction through the LAC-Z gene. You then ligate, clone, and transform, and you will do a count of blue versus white colonies and create some sort of calculation as to the number of errors that were introduced that disrupted expression of the LAC gene. Another way that you can do it is to actually do Sanger sequencing. It's a very tedious process, but you could actually do a PCR and then just do traditional cloning and sequencing of that. And the newest way is to actually use NGS. It's very powerful technology, and you're actually able to generate a lot of sequence information in a fairly quick time. So, and basically you do the same that you would do PCR across a specific region and then sequence it using NGS. So when you're looking at error rate, you actually are looking at the number of mistakes or misincorporated nucleotides within the total number of nucleotides that were polymerized. So in some cases, the error rate for a low fidelity polymerase, such as TAC, will actually be 1 in thousands of bases amplified, versus a higher fidelity enzyme could only make a mistake in 1 in several million bases amplified. So, but because TAC DNA polymerase has no proofreading activity, we use that as our baseline for all other enzymes that we measure against. So in this image, we've taken TAC as our baseline and set it at 0. And then we've taken all other high fidelity polymerases, including our new Invitrogen Platinum Superfy polymerase, and measured the fidelity of that using NGS and compared it to the fidelity of TAC. And in this image, as you can see, there's a lot of high fidelity polymerases that will give you, on average, between a 15 and maybe a 30-fold increase in fidelity over TAC. But there are some other high fidelity enzymes on the market that will give you much better improved results. So other key factors that you want to consider when you're doing high fidelity PCR reactions is to obviously use the enzyme and the buffer and the reaction conditions that are supplied with the enzyme. Typically, this is important because salt concentrations can actually vary between the different enzymes, as well as concentration of enzyme, then making sure that customers will commonly add some sort of an additive like DMSO to their reaction to potentially get through any high GC content regions. And we've actually documented that the addition of some of these additives can actually impact and decrease the fidelity of the polymerase. Another key feature is to make sure that you're using the highest quality DNTPs so that you're not getting any challenges due to degradation. And also to limit the number of cycles. Typically in a standard cloning reaction, you generally only want to do about 25 to 30 cycles of PCR rather than the commonly 35 to 40 cycles. So you want to make sure that you're minimizing the risk of potential introduction of mistakes. So you obviously, because you're doing fewer cycles, you want to make sure that you're using a polymerase that has a high level of processivity to be able to make a lot of product. So this just demonstrates how the impact of a high fidelity enzyme can impact your results of a cloning experiment. As I stated earlier, TAC polymerase has no proofreading activity, so the elongation time is about 60 seconds per kilobase. So, and because it has no proofreading activity, if you were to amplify a piece of DNA and clone it, on average, about one out of every 2 colonies picked would have an error introduced into them. Another high fidelity polymerase that's on the market is PFU. It has an 8-fold fidelity increase over TAC, but as you can see, because it is a true proofreader, it has a very low processivity rate because it's very slow as it wants to make corrections to any mistakes within the DNA synthesis. So what we find, though, is about 1 in every 4 clones can potentially have an error. And this was all done with a 3 kb fragment and a 30-cycle PCR. We did the same experiment with platinum superfi, which has a fidelity of greater than 100x, and it's actually been engineered to be very, very processive. So you can actually synthesize about 15 to 30 seconds 1 kilobase of DNA. So you're able to decrease the extension time in your PCR reaction even for longer templates. And because of the higher fidelity, we've actually found that less than 1 in every 50 colonies picked had any kind of an error. Another key thing that people will be doing where they want to make sure that they have the most accurate polymerase present is to do site-directed mutagenesis. There's typically 3 ways site-directed mutagenesis is done. If you want to incorporate a point mutation into your DNA strand, you will design a primer containing that point mutation. You'll do some amplification of this plasmid, then you'll ligate and clone so that that point mutation is actually incorporated into all of the DNA that's synthesized. Another experiment might be to do a deletion. Maybe there's a region of DNA that you would like to exclude from your target plasmid. So you'll actually design primers that will delete the desired sequence that you want to remove. Again, do PCR, ligate, and clone. And the last is maybe you want to actually insert additional sequences into your plasmid. So you will design your primers, and actually on the 5' end of one of your primers, you'll add additional sequence. And again, PCR, ligate, and clone, such that you are inserting that new sequence into the overall sequence of your plasmid. In this experiment, we actually wanted to test the efficiency of superfi in doing a true site-directed mutagenesis experiment. So we started with a plasmid that actually contains a mutation in the LAC-Z gene. We designed primers that actually overlap by 15 bases so that we would introduce the wild type sequence within that overlap. We did our amplification of 30 cycles, and then we transformed it into competent cells, and we did a blue-white screen. And from those results and the number of blue versus white colonies that we got, we actually were able to prove that we restored fully functional LAC expression as compared to wild type. Another concern that people have when doing any kind of PCR, especially with high fidelity enzymes, is we're concerned about the 3' to 5' exonuclease activity being active prior to initial heat denaturation in our PCR reaction. So we used the same methodology that we used to create our hot start enzymes, and we developed an antibody that actually bound the 3' to 5' exo activity until you do a 15-second denaturation prior to your initial round of PCR. So that way, not only so there's 2 enzymes or 2 antibodies actually bound to our enzyme. One blocks DNA synthesis by blocking the DNA binding domain, and the second antibody blocks this exonuclease activity such that the enzyme is completely inactive until that initial denaturation step. Because what we did discover is if this exonuclease activity is left unprotected prior to PCR, and you actually leave your reaction on a bench, that exonuclease activity could potentially hydrolyze the primers that you're going to be using in your PCR reaction, which would create either no amplification or nonspecific amplification due to the degradation of your primers. So another key thing that we wanted to be able to do is show how this enzyme performs across a broad range of amplification lengths. So first, in this first image, we've taken several proofreading enzymes from several different companies, and we've amplified it across a range of different sizes. We had from 0.2 kb up to 20 kb, and we did a standard PCR amplification, and you can see that you actually are getting a lot of non-specific amplification and smearing. So the proofreading activity and the hot start capability of these enzymes is actually suboptimal. We did the same experiment using genomic DNA with platinum superphi, and you can actually see at each size range of amplification, we're actually getting a very clean PCR product. Another concern that researchers have is either high AT-rich or high GC-rich content in their template. So in this, we actually amplified an increasing percent of GC content using platinum superphi polymerase, and you can see even as we were getting to 76% GC content, we actually were getting very, very clean PCR. As I discussed hot start earlier and using an antibody-mediated hot start, we wanted to be able to test how long you could actually leave a reaction at room temperature and still block polymerization or synthesis of the DNA polymerase. So in this experiment, we set up a reaction amplifying a 500 base pair fragment from human genomic DNA. We started with 50 nanograms of starting material, and we did a series of competitors' polymerases as compared to in lane P, which is the platinum superphi. In the 0 hour, we set up the reactions and we actually loaded them directly into the thermocycler and did our PCR. In our second reaction, we had a replicate set of samples, and we actually left those samples sitting on a bench top at room temperature for 24 hours and then did our amplification. And as you can see, in some cases, you get no amplification from the competitors' polymerases, but with ours, it's a very, very clean fragment. We've actually repeated this experiment going up to 72 hours and got similar results using platinum superphi. So now that we've talked about key attributes of a DNA polymerase, now we would actually like to show how we can use those attributes in our real experiments. So first, I wanted to give you a little bit of explanation about how we actually engineered this polymerase. So this yellow image here is the DNA binding domain of the polymerase, and we actually genetically engineered this using some fusion chimeric proteins to create an enzyme that not only gave us better processivity that would give us improved yield when we did our amplification, but we also wanted to make sure that it was fast, that we were able to do long PCR. We also wanted to make sure that it was resistant to inhibitors such as heparin, because a lot of researchers now would like to be able to start directly from their material rather than doing a purification step. And then, as we demonstrated earlier, we also wanted to show how it can perform in GC-rich environment. So as we stated earlier, when you have a low processivity enzyme, such as a true proofreader, because it's constantly having to go in the 3' to 5' direction to check for any potential errors, it actually slows the overall synthesis down, so you don't get a lot of product on your gel due to this impact of the processivity. Typically, a high processivity enzyme will actually speed through and synthesize a lot of DNA, resulting in very, very nice fat bands on your gel because of a lot of PCR product. One thing to note, because of the way in which the DNA binding domain of this polymerase was generated, it does actually impact the melting temperature of the binding and annealing of your primers. So we've actually developed an algorithm that we have directly on our website where you can take the sequence of your primers and just copy and paste them into this calculator to apply the algorithm to make sure that you're actually getting the ideal annealing temperature when programming your thermocycler. You don't actually need to redesign the primers. You can just take your existing primer sequences and put them into this algorithm. And typically what you'll see is about a 2 to 3 degree increase in the annealing temperature that you would normally use when you use a Taq-based polymerase. So when we were talking about starting material and concerns about inhibitors that might impact the performance of the PCR enzyme, we wanted to show some key inhibitors and how this enzyme or competitors' enzymes might be impacted by these inhibitors. So in lane 1 of each of these experiments, there's no inhibitor. We're actually amplifying a 2 kb fragment from genomic DNA. In lane 2, we've actually, and A, B, C, and D are actually competitors' polymerases. And in each of those reactions in lane 2, we spiked the reaction with heparin, which is commonly found in blood. In lane 3, we spiked xylan, which is actually commonly found from plants. And in lane 4, we spiked humic acid, which is a common inhibitor found in soil. And as you can see, with platinum superphi, in all of these cases, you're not seeing any impact or inhibition to the result of your PCR reaction, whereas with the other competitors' polymerases, you can greatly impact the success of your PCR. We demonstrated earlier how platinum superphi can perform in the presence of increased amount of GC content, but we actually wanted to also demonstrate how it compared to other competitors' polymerases on the market. So in this experiment, we actually did a PCR with increasing amount of GC content going up to 76% of a standard target from human genomic DNA. When we actually created a new GC enhancer that's a buffer additive that you can actually add to your PCR reaction, and you can actually titrate it to be used when you're working with samples that are about 70% or higher in GC content. So in this particular reaction, we did actually add that GC enhancer to the solution to make sure that we're melting those structures to get successful amplification. When you're working with long PCR, when you're actually going to be amplifying fragments that are greater than 10 kb, then there's some other challenges that you may actually face. As we talked about with the processivity of the enzyme, if a proofreader has a low processivity, then it actually has challenges being able to navigate and create longer fragments. So that can actually create a lower yield or lower amount of product on your gel. Another concern, just like with the number of cycles, that you want to minimize the number of mistakes when you're doing long PCR, because your error rate has to do with the number of bases synthesized, then doing a longer PCR could potentially put you at risk to introduce additional errors or mistakes. And also, longer sequences have a tendency to be complex in their DNA structure and makeup. So in this slide, we wanted to be able to apply that thinking to a real experiment. So we had a 6 kb sequence that we wanted to be able to sequence using RT-PCR. So the first thing that we did was we isolated RNA, total RNA, from mouse liver, spleen, and kidney. And because tissues are very, very high in RNase, we wanted to make sure that we weren't potentially degrading our RNA samples prior to us doing cDNA synthesis and PCR. So in this image, we actually added an additive called RNA later to preserve the RNA integrity in our stored sample. And we stored these samples at 4 degrees for 1 month. And as you can see from the image, we actually still have very, very intact 28 and 18S RNA, so that we know that we actually were successfully able to block the RNase activity from degrading our samples. So next, we actually wanted to do our cDNA synthesis for our 6 kb fragment. So from this particular image, we used a variety of different reverse transcriptases to do our cDNA synthesis. And actually, as you can see in this middle lane, this is superscript 4 reverse transcriptase, which is a new RT that we've developed about a year ago. And you can actually see that you get really nice cDNA synthesis in that 6 kb range. So we purified this band, and then we did PCR, and you can see in this image where the PCR is. So again, you're getting nice, clean amplification. And then we purified this band and we cloned it. So we were very successful in starting with tissue and actually getting our 6 kb fragment to clone. So as discussed earlier, there are some key tips that you want to remember when doing long PCR. So you want to make sure that your template is pure and fresh and is fully intact and it hasn't been degraded in any way. And again, using fresh primer solutions, so you're getting very, very specific binding and annealing of your primers. You do want to do you might want to do some changes or adjustments to the thermocycling conditions of your polymerase. We actually, in our protocol for Superfy, give you simple instructions for doing up to a 20 kb fragment. But again, just make sure that when you're doing your PCR that you're taking into account the length of the fragment that you'd like to amplify, as well as the extension rate of the enzyme that you're using. And then also just make sure that your polymerase is really, really processive so that you're actually able to find the band on your gel to do any downstream applications. So in this image, we actually wanted to test about the length of DNA that we could successfully amplify using Superfy or, again, our keyers. So in the first image, we took genomic DNA and we are amplifying a 15 kb fragment. So, and you can see that you, again, get very clean amplification with Platinum Superfy, and you get some mixed results with some of our competitors. We also did some minor adjustments to the annealing and extension times of the reaction and amplified, and were successfully able to amplify a 30 kb fragment. We've even been able to do a 40 kb fragment from E. coli genomic DNA. So again, with some minor modifications to the overall extension and annealing temperatures, but we have been able to get the enzyme to go up to 40 kb successfully. When we discussed inhibition, we also wanted to talk about sample type and be able to test the effects of the processing of samples in our PCR. So one common area of work is working with formalin-fixed paraffin embedded samples, or FFPE samples, only because the process of doing FFPE treatment of tissues is very harmful to the DNA in that sample due to all of the heating and chemical treatments that take place. So most of the DNA found in FFP samples is actually probably not the best quality of DNA for doing PCR. So in this set of experiments, we actually took FFPE samples and we did varying quantities of starting material from 0.5 up to 10 nanograms of starting material, and we were able to successfully amplify a 600 base pair fragment. Another key thing that people would like to be able to do is to start directly from blood and be able to do your PCR. And unfortunately, hemoglobin is an extremely challenging reagent in that it will inhibit most polymerases. So, but we were actually able to take a few microliters of blood that have either been treated and stabilized in EDTA or from heparin and actually be able to get successful PCR directly from blood as well. And we've gone so far as to actually even create an application note around the work that we've done with blood. So some of the key highlights that we've talked about regarding Platinum Superfi are that it has a 100-fold increase in fidelity. It actually, we think that it may actually be higher than that, but due to the background levels, we're continuing to sequence additional clones to make sure that we can more accurately confirm what the true fidelity rate is as compared to TAC. It's obviously very high in specificity due to the antibodies that we have bound to the DNA binding domain, as well as the 3' to 5' exonuclease component. We get very robust amplification from difficult to amplify targets, whether they come from blood, soil, or have very, very high GC content. And then again, the benefit of that hot start capability is that you're able to do a room temperature setup of your reactions. And then we did also, just for convenience, provide the sample or the enzyme with a green loading dye if you do in fact want to just do direct loading onto your gel. So in conclusion, with the work that our research group has done in the development of Platinum Superfi, as well as the experiments that we've been able to perform, we hope that we've been able to show how leveraging precision and power to overcome your challenging templates will allow you to achieve the highest success in PCR. Thank you.